Good morning, everybody, and welcome to the Science of Suspended Animation in Deep Space workshop organized by the Translational Research Institute for Space Health, or as we call it, TRISH. My name is Emmanuel Urquieta, and I am the Deputy Chief Scientist at TRISH. Today's workshop will have some of the world's leading experts in the field of torpor and hibernation. Our institute is always looking at novel and disruptive approaches to solve the health and human performance space biomedical problems for future missions to Mars and to deep space. This workshop aims to landscape the state of the art in research related to torpor and hibernation as our institute explores new potential areas of interest. Today, we'll embark into the amazing world of torpor hibernation, or as we call it in this workshop, suspended animation. The field of hibernation is extremely vast, although there's only a small community of investigators from which we will be hearing today. A myriad of approaches and unanswered questions exist in this field. And as we keep learning more about natural hibernators such as bears, lemurs, and other species from some of the ones you will be hearing today from the panelists, our interest keeps increasing to understand if and how we can take advantage of some of the unique physiologic mechanisms that allow these animals to endure such extreme environments and conditions. Some of them are analog to the ones that astronauts will have to endure during missions to Mars and to deep space. Today, we will bring scientific facts, and like the title of this panel and this workshop is, we will separate the science from the science fiction of suspended animation. But before we get into the agenda, and before I provide an overview of the workshop and aims, I would like to thank all of the people that made this workshop possible, but especially Rachel Dempsey, our Communications and Education Officer. She has helped tirelessly all of the last month and so to make sure that this is working today. I also want to thank Dr. Gary Strangman, one of our innovation specialists detailed at Boston, who will be helping to read questions today. I will be providing logistics regarding Q&A in a few minutes. But before doing that, and without further ado, I would like to introduce Dr. Dorit Donoviel, our director, who will give an overview of our institute and will say a few words.
Thank you, Dr. Urquieta. It's a real pleasure to be here today. I am going to attempt to find my slide deck. Okay. Hello, everybody. So welcome from Houston, and I'm really thrilled to hear what you all have to say today. We invite you to engage with us through questions, and we're really very much an open platform for bidirectional exchange. So I'm an associate professor at the Center for Space Medicine at Baylor College of Medicine, and I have the distinct pleasure and honor to direct TRISH, working closely with the NASA Human Research Program. We are a consortium of Caltech, MIT, and Baylor, and we are here to really help NASA continue its journey beyond what it's been doing so well for the last 20 years, which has been working and living in low Earth orbit. For the last 20 years, the ISS has been home for hundreds of individuals, international partners, etc. And as many of you know, we've set our sights also with international partners to go back to the moon and to establish a presence in a laboratory orbiting the moon and go back and step on the moon's surface. And this is all in preparation for the journey that will take us beyond what is in the close vicinity of our Earth and go into deep space, which will be a very long duration mission. And so what you see over on the right is an artist's conception of what an orbiting laboratory would look like on Mars, for example. And we set our sights to help NASA get there. And the Mars mission really kind of changes the paradigm off. But I think I have to leave the details as to the challenges and deep space destinations to Dr. Urquieta. I know he has a nice slide deck to explain some of these. And so he asked me to focus on. And so with that, basically, I just it's easier than the mouthful that is the long name. We are funded by NASA completely and wholly. We're partnered very cooperative. And it's a wonderful relationship because there's a lot of there and find and enable safe, productive human space exploration. And not just on ISS or on the moon, really change paradigm, and Dr. Urquieta will get into that. And also working closely with our distributor, we even have in the UC system, really, so we've been working actually for doing so. So how do we execute our mission? We challenges that for itself in exploring deep space. NASA is a wonderful organization. It has a safety first culture. That means it's much more difficult for NASA to take more, although it puts pocket, it takes a very and courageous organization. But when it comes to conservative, just much like the healthcare industry itself today. And so what Trish is supposed to be doing is more of a skunk works, if you will, out of the box thinking to bring forth some new ways to think about the same challenges that NASA is trying to solve itself. And so the way we do that is really first and foremost through funding research. We have solicitations that we put out. They're open to all US-based institutions. Our average annual grants are multi-year grants that go anywhere from $400,000 a year to $1 million, which just recently closed for $1 million a year for 3 years. There is one that we put out that is focused, and it's going to be on a rolling basis going forward, that's open to companies, to just industry, that because we realize that industry or companies really don't write proposals very regularly, and so we wanted to tailor something that was more convenient to them and easy for them to engage with us without having to write massive proposals like most academic researchers do. We also have an open year-round Catalyst program, which ideas that come forth from the community can come forward. Typically, they tend to be smaller dollars for this, proof of concept studies. So if you have a harebrained idea that you think we really should know about it and could really help NASA, bring it forward, and we're probably the ones that are going to give you that first bolus of money to make your dream a reality. And we also have some directed projects, which we have a need, and we will search out the best people to really custom make a solution for NASA. We do that on a less frequent basis. We generally stay open to your ideas, but occasionally there is some things that just absolutely need to be done according to specs. And so these are all the different ways that we can fund research. We are also very much engaged with workforce development. What I mean by this is investing in the people. It's a bidirectional education of, and also a support of salaries for postdoctoral fellows, for example, for medical fellows. We have a surgery fellow that's currently working at UCSF that is also understanding what it takes to be a medical person in space. We also have this wonderful program called the Space Health Swap, which you can spend some weeks at NASA centers throughout the nation and learn about what NASA's needs are, and maybe begin a collaboration with some of the NASA researchers, the intramural researchers. And then we also have opportunities for NASA researchers to go out and spend time in your labs. And so we've initiated this program in 2020. It was really successful, and we were about to place people, but unfortunately the pandemic hit. So we're probably going to open up this again, and it's really a wonderful way to engage with the space program and learn more. I mentioned already the bidirectional exchange of information. We provide a lot of scientific content that is archived on our website. We learn from you, and you learn from us. And so this is largely like this workshop today is one of them. We last month we had a wonderful workshop on the microbiome that was hosted by Caltech and Trish, organized by some of our folks with Steve Mayo, who's a scientist who's a part-time person that works for us as well at Caltech. And Rachel Dempsey, our communication education lead, has been organizing the Red Risk School with the help of Jeff Willey, an assistant professor, sorry, I think he's an associate professor now at Wake Forest. And the Red Risk School is all around what the red risks are, which NASA classified with the color code red being the most impactful for deep space missions. And so all of those lectures from NASA experts and Trish experts around NASA's needs in deep space are archived on our website. And so we invite you to look at some of these incredible resources for scientific information. But the idea here is that we really kind of help solve these health challenges together. And while we are solving for deep space, we really are making an impact here on Earth, because the paradigm that we set for deep space missions, for healthcare at least, really has a lot of applications for today's healthcare system. So the workshops I mentioned, the in-person conferences when we are all able to get together, we do have at least one of those a year. We have our Orbit, which is our wonderful community, which Rachel monitors and organizes, where you can sign up and connect with others and have content and engage in conversations and discussions with people that have shared interests. We also have monthly newsletters, which we tell you about funding opportunities, and we have monthly meetings, and we invite everybody to take part. So if you'd like to join us, please do so. There's lots of opportunities to do that. So you can be part of our Space Health Orbit. You can apply for funding or our workshop development opportunities. You can send us ideas for topics for research, and we will definitely consider them. We often do workshops around topics that we are thinking about putting out a solicitation for. So we will probably have a solicitation coming up. That's our normal cadence in the fall, which we're coming up to. And since we just had a workshop on microbiome and a workshop now on suspended animation, it doesn't take a rocket scientist to figure out that we're interested in these areas. You can also apply or tell us that you're interested to be a reviewer. We really need out-of-the-box reviewers. So when we get out-of-the-box proposals, there's nothing worse than hearing a conservative reviewer kill it because it's absolutely groundbreaking and they don't believe it'll work. So if you are an out-of-the-box thinker, come forward and be a reviewer for some of our out-of-the-box proposals. Participate in the conversations and the workshops, and then let others know. Bring in your colleagues, and we really are hoping to engage as large a community as possible in what we're trying to do for NASA. And with that, I'll just leave you with a couple of links. Feel free to reach out to me or Dr. Urquieta or any of us, and we're always open to have a conversation with you. Thank you so much for this opportunity, Dr. Urquieta, and I'll send it back to you.
Thank you, Dr. Donoviel. Okay, I appreciate that introduction of Trish. Now we're gonna talk a little bit about setting the stage. What we want to do with this workshop. What do we want to accomplish? I'm also gonna go through questions and answers. How we want to do questions and answers from all of the attendees, and then I will read the bio sketches from our amazing panelists today during this part. We have 30 minutes for each panelist. We're going to have 25 minutes for presentation and then 5 minutes of questions and answers. So what I want to do during the last 15 minutes that I have during my section here, I'm going to introduce each of them so that we don't have to spend that time during their presentation. They can really focus on their topics. So like Dr. Donoviel was saying, we have been on low Earth orbit for quite some time. And the challenges that we have been tasked to solve or to help solve from the Human Research Program are very different from the things that have been solved and the things that happen on the space station. We have been on low Earth orbit for the past 20 years or so. Pretty much the astronauts remain healthy. There haven't been any evacuations from astronauts from ISS. Some of the issues that used to be problems before, such as loss of muscle and bone, there's very mature and very good countermeasures to help them to recover some of that muscle and bone that get lost, given that they have no gravity in space. So we take these things for granted, but where we are going next is very, very different from low Earth orbit. We're talking about millions of miles, not only 250 miles or so, which is the altitude of the space station. Um, a traditional mission on space station is 6 to 12 months. A return to Earth takes only hours. I'm pretty sure that most of you saw the landing of the mission 2 from the Dragon mission 2 last Sunday, and it was really exciting. We're really getting into a new era of exploration. However, there's two phases after low Earth orbit. And the next one that we're all familiar with is the Artemis mission that will take us to the moon in the 2024's timeframe. And these are missions that are going to be similar in duration from the ones that we have on ISS. These ones are going to be 1 to 12 months. However, the return to Earth will take days, 3 to 4 days. And if there's an emergency or if there's any concerns or any issues there, it's not that easy to return. And this is going to be the test bed for Mars. Now, Mars mission is a completely different story. This is something that will take 2 to 3 years. Return to Earth will take months or pretty much a whole year, and there's going to be no communications. The space where astronauts were going to be living, the physical space, is absolutely small. And there's going to be 4 astronauts on this very, very small capsule. So how can we solve those huge challenges, those huge problems from novel ideas? What investments can we do today that will have an impact in the future? How can we think out of the box, non-traditional solutions? How can we de-risk novel and like Dorit was saying, risky problems, risky solutions, so that we can have an impact in the 2030s timeframe when we're doing a mission to Mars? And that's something that we will do today. We will have starting a conversation around torpor and hibernation. One of the multiple challenges that we have for a mission to Mars is, of course, the environment, the radiation environment of deep space. This is galactic cosmic radiation, is very high energy radiation that is very difficult to shield. The environment is also very challenging. We have mildly elevated CO2 on the spacecraft. We have low humidity. We have issues with the circadian rhythm. Of course, the astronauts are not going to be having 24-hour days like here, so they're not going to be having dawn and dusk. So all of these environmental challenges have to be considered for these missions. The other one is isolation and confinement. Imagine that you are with another 3 folks that your boss chose them. They're not necessarily your best friends. So you're going to be with them stuck in a very small capsule for 3 years. There's going to be probably no windows, and you cannot go to a corner and isolate yourself if you have any argument with anybody else. So how can we make sure that we have countermeasures and keep them happy and healthy, not only from a physical perspective, but also from a mental and behavioral perspective? So this is other of the huge issues that we have for I won't call it issues, but challenges that we have for deep space missions. And the other one that sometimes it's difficult to conceive is the distance from Earth. If you remember the Apollo 8 mission, it was the first one that went around the moon. And there's this very famous picture from one of the astronauts when instead of us looking at the moon coming from the horizon, they actually saw the Earth coming from the horizon. And that was really challenging because it was the first time that somebody saw such a big distance from Earth. Now, this is a picture that you see here that was taken from Mars from one of the rovers. And that little dot that you see, there's not a star. That's actually the Earth. So that's the other huge issue. We'll have distance from Earth that will have repercussions from communications and other aspects. Time delay communications. We cannot rely on just grabbing the phone and calling mission control in Houston. And if there's a medical issue or if there's a behavioral issue, we cannot have real-time communications with them. It's going to be taking around 20 to 40 minutes to get those communications back and forth between Mars and Earth when both of them are at the farthest distances. And having said this, one of the ways that we try to approach these challenges is considering them as 5 hazards of human spaceflight. One of them, like I was saying, is distance from Earth. This one encompasses communications delay, autonomous medical care, and no emergency evacuation. If there's a medical emergency, of course, they cannot just turn 180 degrees and come back to Earth. Once you go to Mars, You have to go all the way there. Then you have to go around around Mars and then come back. So if there's a medical emergency, there's no real-time possibilities for evacuation. Isolation and confinement, I was mentioning the behavioral aspects and also the sleep disorders. Then we have the hostile closed environment, and here we have the vehicle design. How can we design the vehicle so that it's good enough and provides a nice living space for the astronauts? The environment, CO2 levels, potential toxic exposures, microbiome. How does the microbiome changes given this isolated effect? How does the microbiome from the spacecraft has any potential implications with the microbiome from the astronauts? And like Dr. Donoviel was saying, a few weeks ago, we had a workshop held at Caltech from Dr. Steve Mayo, uh, really talking about microbiome, the host microbiome, and how even potential things as the gut microbiome axis could potentially be used for spaceflight. And then we have food nutrition. How can we provide food and nutrition that are adequate for these astronauts? On the other hand, we have radiation effects. As I was saying, the radiation on space is galactic cosmic radiation. It's very difficult to shield and it's very different from traditional radiation that we encounter in terrestrial environments from nuclear power plants or from imaging procedures in hospitals. And from these, there's three main areas that we try to focus from its effects. The first is the acute effects that we'll see on the in-flight portion of the mission. Then we see the long-term effects that we'll not see during the mission, but after the astronauts come back, potentially cancer. And then the acute effects from chronic but low doses of radiation in the central nervous system and the cardiovascular system. And finally, we have the altered gravity fields. Disorientation, balance disorders, fluid shifts, cardiovascular deconditioning, exercise, and fracture risk. So how do we approach these challenges? We, in the past, have tried to approach these from a multi-species perspective, trying to understand at different levels, and then at the end coming in a human analog or a human-like model. And this is one example. We can start understanding problems from cell cultures, from bacteria like E. coli. Then a next level will be something sort of like a tardigrade, a water bear. Then we try to understand these approaches or maybe some genes that we found here in flies or other species that are a little bit higher in the evolutionary perspective. And then we can have humanized mice or other types of models that have been going through different levels of validation. And finally, we're looking now into having human chips or human chips models or microphysiological systems that have actually human cells and can actually be put into an environment that simulates the effects of spaceflight. And of course, at the end of the day, we want to bring this back to the astronauts to make sure that they can go back and forth from Mars safely. One of the examples that we have had in the past, one of our funded projects was looking at water bears. Then the genes that were found there that could provide potential radiation protection, genes that are homologs to water bears in humans, they were cloned into flies. Then from there, they could go into transgenic flies, then they could be irradiated, and then we can come up with a library of genes that are human homologs for different tardigrades and flies and humans. And then we have a library of genes that we can overexpress potentially in the future, providing a new way for giving countermeasures to these astronauts. So in the same line of thought, we try to have all of our projects multi-approach, sort of like a Swiss Army approach. And this is what we'll be discussing today with hibernation. These are using non-traditional solutions and non-traditional countermeasures. We need to maximize the resources that are extremely constrained from a mass, power, and volume perspective in deep space missions. Now, I'm sure that some of you, when you registered for this workshop, this is what you thought that we were going to discuss today, but I'm sorry to tell you that this is not what we will be talking today. This is what we usually see in the movies. This is what we think of when we talk about suspended animation and hibernation and torpor. I'm sorry to tell you that we are not planning to freeze astronauts. We're not planning to create capsules like this for deep space missions. What we're trying to achieve today is understanding how different natural hibernators, such as these beautiful animals that we see here, and we have the world's leading experts on each of these animal models, how can we find the genes that they have? How can we find the pathways that they have? How can we find the hundreds or thousands of years of evolution that these animals have had to endure, so that, for example, a bear during hibernation loses a lot of muscle, but then during a pre-arousal period, this bear really starts producing muscle again. So how can we take advantage of these natural capacities, these natural capabilities of these animals, and how can we translate these ones into spaceflight? How can other animals, for example, have neuroprotection? How some animals have really increased reaction oxygen species scavenging? How can we use these to reduce inflammation or maybe to potentially protect astronauts from radiation? This is what we'll be talking today. And going back to that same idea, we will be separating again the science fiction from the science of hibernation and torpor. And our end goal is to find a point of commonality between all of these different approaches, so that we can have again a Swiss Army approach. All of you have seen the agenda. We have some morning sessions. Then we have a 30-minute break at 12:30 Central. You can go and grab your lunch. You can have bio break, and then we'll come back at 1 p.m. for the afternoon sessions. And then at the end of the day, from 2 to 4, we'll be having all our panelists. We're going to have Dr. Christine Fab, our chief scientist, and myself moderating and having a panel discussion on what we should do in the future. How can we make sure that this field helps the spaceflight, the human spaceflight field in the future? So we'll be having really interesting discussions during that time. Now I'm going to go and introduce each of our amazing speakers today. Our first speaker today is Dr. Sandy Martin from University of Colorado, and she will be talking about evolutionary genetics of hibernation. Dr. Martin holds a Ph.D. from UC Berkeley and is currently a professor of cell and developmental biology at the School of Medicine of the University of Colorado. After her PhD, Dr. Martin became interested in hibernation during her postdoc, where she heard a faculty colleague at the University of North Carolina, Chapel Hill, speak about this work on hibernation induction triggers. She found hibernation fascinating, wondering how some mammals could orchestrate and survive such a profound deviation from what was understood to be mammalian homeostasis. From an evolutionary perspective, the distribution of species that hibernate suggests that there's a common mammalian ancestor that was a hibernator. And I'm really interested to see what she means by this and how we can discuss this during the panel. It has been the goal of her lab from the beginning to define the molecular mechanisms underlying the hibernation phenotype. Our next speaker is Dr. Kelly Drew from the University of Alaska Fairbanks, and she's going to be talking about hibernation neuroprotection. As you can see, all of our speakers were chosen based on their areas of research and how these specific areas can go back and help us solve the problems that I showed at the beginning, and how can we find a commonality on all of them. Dr. Drew studied neuropharmacology at Mount Sinai School of Medicine and at the Karolinska Institute before returning to Alaska in 1990, where she is now professor of chemistry and biochemistry at the University of Alaska Fairbanks. She's passionate about the neurochemistry of consciousness. Dr. Drew fell in love with hibernation. Over the past 25 years, Dr. Drew's research group discovered unprecedented neuroprotection in the hibernating brain and a mechanism necessary and sufficient to drive the onset of hibernation. She founded Be Cool Pharmaceutics LLC in 2015, who is looking to commercialize therapeutics for rural or remote emergency medicine and human spaceflight based on hibernation. She also founded and directs the Center for Transformative Research in Metabolism at the University of Alaska Fairbanks. The center aims to translate hibernation to improve human health. Our next speaker is Dr. Matteo Seri. He is an MD PhD from University of Bologna, Italy, and he's going to be talking about hibernation and radio protection. Dr. Seri is an MD PhD. He completed medical school at the University of Bologna, and he has a PhD in neurophysiology from the same institution. Dr. Seri served as a medical lieutenant in the army in Italy, and his research mainly focuses on physiology of hypometabolism, torpor, hibernation, and the related effects and the derived applications, including but not limited to radiation resistance, synaptic plasticity, aging, control, or food intake. Our next speaker is Dr. Kenneth Story from Carleton University in Canada. Dr. Story will be talking about metabolic and rate regulation as it relates to hibernation. Dr. Story is a professor of biochemistry at Carleton University in Ottawa and holds the Canada Research Chair in Molecular Physiology. Dr. Story graduated from the University of Calgary and the University of British Columbia. Dr. Story's field of interest is biochemical adaptation. In particular, mammalian hibernation and freeze tolerance are core topics in his over 900 publications. His expertise in the natural mechanisms of hypometabolism and freeze tolerance has frequently been put to use by cryomedical and organ preservation research groups. Then we have Dr. Matt Reagan from University of Wisconsin-Madison. He will be talking about hibernation gut microbiome. Dr. Reagan is an NSERC postdoctoral fellow working with Dr. Hannah Carey at the University of Wisconsin-Madison. He's a comparative animal physiologist whose work investigates metabolic depression used in different vertebrate groups. Additionally, over the past 3 years, he has worked with colleagues at NASA and Jacobs Engineering on projects related to applications of synthetic torpor to long-duration human spaceflight. And last but not least is Dr. Claire Nordin. She is an ER physician working at the Bartlett Regional Hospital in Juneau, Alaska. She will be talking about the terrestrial uses and the state of the art of clinical targeted temperature management. Dr. Nordin is a physician board certified in emergency medicine and emergency medical services. She completed medical school at the University of Pennsylvania, residency at Denver Health, and finished her training with the University of Washington EMS Fellowship with a certificate in the Teaching Scholars Program through the Department of Bioinformatics and Medical Education. Dr. Nordin lives in Bozeman, Montana, where she will join the group of emergency physicians at the Bozeman Health Diaconus Hospital this fall. Before we start, we are already getting into the 10:30 timeframe. As you can see, our speakers are absolutely amazing, and I'm sure that we'll have a lot of fun, but more importantly, we'll learn a lot about the field of hibernation and torpor. Just some logistics. Please use the Q&A button that you see on the bottom, the icon, only for questions. If you want to ask a question to the panelists, please make sure to send it in the Q&A and not in the chat. If you have any technical issues, that's what we have the chat for. After the panel, during the 30 minutes, the last 5 minutes will be used for questions and answers, and Dr. Gary Strangman will be reading the questions to the panelists. Some selected questions that we don't have time to answer during the 5 minutes, during the 30 minutes, and then the 5-minute sections will be considered during the last part of the workshop, during the panel, during the 20 or 30 last minutes. So please have fun and learn. For our panelists, we have exactly 30 minutes for each of you. Please, at the 25 minutes mark, I will remind you that the time is over, and then we'll take questions and answers. So with that being said, our first speaker today is Dr. Sandy Martin. So Dr. Martin, if you can share your screen, the stage is all yours.